Welcome to Synapsis. I'm Gökçe Özdemir, a biomedical scientist specialized in neuroscience. In biology, synapsis is a stage during cell division where chromosome pairs line up before exchanging genetic information. For me, synapsis is a bridge to make medical science more understandable for everyone. Thank you for being here. Let's dive in. Today, I'll be talking about stem cells, the cells that create all tissues and organs in the body, a topic I've personally worked on for years. The current popularity of stem cells comes from their potential to treat diseases, whether it's regenerating damaged organs or building new ones altogether. The key definition of stem cells is their ability to replicate themselves, continuing to produce identical copies unless their environment changes, which can force their differentiation, meaning turning into specialized cells that commit to a certain type and can no longer make new copies. After a sperm fertilizes an egg, one single cell's DNA is replicated into trillions of cells. In this journey from one to trillions, there are countless clues that can improve our health. And as new clues emerge, so do new myths. The purpose of today's episode is to debunk a few myths. Before we get into the myths, for the sake of the first episode, let's do a quick introduction. I was born and raised in Turkey, and thanks to a family of physicians, I grew up fascinated by the human body. And the more you explored, the more questions arose. So after moving to Canada, I did my undergrad in life sciences, and with summer internships in the US, I started specializing in neuroscience. Then I pursued my master's degree in Toronto, continuing on the neuro path, focused on stem cell therapies for spinal cord injuries. Later, I continued in the biotech industry, focusing on rare genetic disorders and genetic modifications. I believe there is a great need for awareness in medical research worldwide, and my goal here is to be a tool to help develop this understanding. The name of the channel, Synapsis, was born from this philosophy. Each egg or sperm cell that you have went through a process called meiotic cell division, during which the chromosomes you got from your mom and the ones you got from your dad aligned next to each other in the synapsis stage, and the arms of the chromosomes quite literally crossed over to exchange genetic information. This way, each egg and each sperm received different combinations of DNA. Science and some luck then decided which genetic combo would create you. Inspired by this system, I created Synapsis, the podcast, to exchange scientific information and bridge the knowledge gap. Okay, now on to stem cells. One of the biggest myths about stem cells has to be that they can cure any disease. While they do hold promise in many areas, the globally recognized and approved therapies are still limited to mainly blood disorders such as leukemia or lymphoma. There are many candidates in the research phase, some in the clinical trials already given to eligible patients. The list of candidates is certainly long, from spinal cord injuries, Parkinson's disease, osteoporosis, to type 1 diabetes or heart disease. These conditions are all caused by the absence or damage of a specific cell type. However, if the stem cells are not designed to meet the specific needs of the damaged tissue, they could even be harmful rather than helpful or not do anything at all. This brings up the question, which treatments are applied, where, to whom, and how? Regulations play a big role here, as different countries have different rules about how much scientific evidence is needed before a treatment can be given to the public. Countries like the US, Canada, and the UK have very strict regulations, which limits the application of stem cells, while in places like Mexico, regulations can be a bit looser, leading to more options and a lot of stem cell tourism. While it's easy to find cases of people who might have benefited from a procedure, unfortunately, there are also many patients who end up not seeing any results at all. Another interesting case here is in Turkey because of its fastly growing health tourism with stem cell tourism in it as well. The applications that are the most apparent seem to be around cosmetic applications or joint problems, but the market has already grown to a point where it's incredibly important to do your research about which promised treatments are based on high quality scientific evidence rather than theoretical approaches. False marketing aside, while there are approved stem cell therapies that exist, It's a fair question to ask why those treatments can't be applied to other diseases. Let's call this the "a stem cell treatment can be applied to many conditions" myth. To understand this, let's understand some of the biology. Just like every disease can have different types and mechanisms, there are many types of stem cells with different functions, different maturations, and different needs. The first stem cell at the top of the food chain is as free as a wild bird. It can turn into both a baby and its placenta and the umbilical cord. This variety makes it a totipotent stem cell. Next up is a pluripotent stem cell, which can make the whole baby, but not its surrounding tissues. After that comes the multipotent stem cell, which is specialized into different systems of a body. A hematopoietic stem cell can make all blood cells, but can't make a bone cell. Or one that makes the lung can't make the brain. What's really fun about this is that every cell carries the same DNA. The only thing that's different is how they use the DNA to make different proteins. Think of a really long recipe book. An early stage totipotent stem cell can choose to make anything from an appetizer to a main dish or a dessert, or even the cover of the book itself. But a multipotent cell can only make a recipe from a specific section of the book. Just like how sometimes a main dish could require something sweet to be added, sometimes a stem cell of one category could help improve the recipe of another category. But what's crucial here is knowing exactly how much and what type of a sweet to use for which other dish to actually improve it, rather than wasting the sugar or ruining the whole dish by adding the sugar. If we want to use stem cells that make up the brain to fix a brain disease, for example, we have to respect the fact that there are about 170 billion cells in the human brain. In 2023, 21 papers were published as a series. Their common goal was to improve our understanding of the brain. The majority used single-cell genomics to do this, meaning they investigated gene expression patterns of cells individually rather than looking at a cluster at once. One paper used brain bits from 75 epilepsy patients, for example. Another one used the brains of 17 deceased children, while another used chimps and gorillas and monkeys. The finding we really care about for the purposes of this episode is that they discovered more than 3,000 subtypes of cells in the human brain, based on which genes they used and how. Considering this incredible complexity of the brain, when we think of giving stem cells to this organ, what type of cells I would need my stem cells to turn into can change drastically. There are multiple questions to ask in this process. From the damage, how many and what type of cells are dead? How much time has passed since the injury? Is there a proper chemistry in the tissue to allow for new cells to thrive and make connections? Can I even design cells that can address this damage? Can I safely do gene editing on these cells to grant them certain advantages? How can I make sure that they will reach the target tissue? If they reach the target tissue, can they stay alive? If they do, can I guarantee that they will turn into the specific cell type that I want? Then, will they even be functional? And if they are, will this functionality actually provide observable benefits? Each of these questions can take years to answer sufficiently. This is why each stem cell treatment has to be pursued under the guidance of published studies that explore the same disease and the same cell type match. Now, moving on to another myth, the idea that stem cells can stop aging. They can do many things, but they can't stop aging. Stem cells can't stop the DNA damage and the genetic changes that come with aging, but they can slow down the progression of tissue damage in diseases like osteoporosis and improve the quality of life. A lot of research is being done to bring these treatments into the clinical practice, but there isn't yet a globally recognized protocol that's proven to be safe and effective. This might make you think of SVF, which stands for stromal vascular fraction. It has become a very popular regenerative approach around the world, and while the technique is still in its research phase, its clinical applications began a while ago. It involves harvesting stem cells from fat tissue, especially for cosmetic and joint problems. I'd call this the SVF is pure stem cells and is approved for safety and efficiency everywhere myth. Fat is collected, cells are separated from the fat itself, and then reimplanted to the patient. But the reimplantation mixture contains various types of cells, with only a small percentage actually being stem cells, maybe as little as 1%, and maximum around 10%, depending on the quality of the process. The benefit of SVF is less about the stem cells themselves and more about the proteins secreted by the entire population of cells in the mixture. There is no doubt that in the near future, SVF can have a very important role for regenerative treatments. But there is an important misconception. Some claim that SVF is approved by the FDA in the US, which is false. The only stem cell treatment approved in the US is for blood disorders, using stem cells from the umbilical cord blood. The reason other techniques haven't been approved is that there hasn't been enough high-quality evidence on their safety and effectiveness. And SVF is in this list of still needing stronger scientific evidence. This doesn't mean SVF didn't exist in the clinics. For a long time, clinics were able to stay away from FDA's radar by arguing that SVF is not a drug. But things started changing in 2017 in Florida after 3 patients who received SVF treatment for macular degeneration completely lost their vision, and lawsuits began. The clinic was found to not be complying with manufacturing guidelines while also advertising this treatment to be a cure for many conditions, when evidence for that didn't really exist. This resulted in a 2019 court decision that barred the clinic from offering any future stem cell treatments. During this period, in 2018, FDA also started investigating a clinic in California, where thousands of patients had already received SVF treatments. While it was argued that SVF is not a drug because it originates from the body, after some back and forth, finally in September 2024, SVF was ruled to be a drug that needs FDA approval. It didn't matter that the treatment was your own tissue. It mattered that the tissue still went through manipulation to separate the cells before it was given back. Now, let's talk about something that was once a fact and is now a myth. The idea that pluripotent stem cells can only be obtained from embryos, meaning from unborn or undeveloped human or animal fetuses. Thanks to Shinya Yamanaka, who won the Nobel Prize in 2006, we can take a small skin biopsy from an adult and reprogram the cells to become pluripotent stem cells. In just 2 to 3 weeks, we can turn these cells into any type of cell, able to replicate themselves and differentiate into other cells as needed. You take a skin cell that has stopped dividing and its only purpose is to serve as a skin cell, and you turn it into a stem cell that can become anything. This technique completely changed the game. At that point of pluripotency, we can either work with pluripotent cells as they are, or produce multipotent stem cells, which are focused on a particular system. We can even create disease models by introducing specific mutations through gene editing. And because stem cells can replicate themselves indefinitely under the right conditions, we can use the cells from one person's skin to conduct research in labs all around the world, reducing variability that comes with using cells with different origins. For example, if you're researching Parkinson's disease, you can create stem cells from both a healthy person and a person with Parkinson's disease. Then you can study the effects of treatments or technologies directly between the 2 sets of cells without needing embryos. Even though the cell changes its type, it still carries the mutations that cause the disease. And now you can make endless copies of it without risking variability that would come from using different sources. This really helps in vitro validation. In fact, most of modern research is started in vitro, meaning cells grown in petri dishes without animal use. Before moving to animal testing, you need to show that the technique works in vitro. And the ethical committees still require solid rationales for why animals are necessary for your study and why the experiment has to be done in the specific animal you're proposing. rather than a smaller, less evolved species, let alone the higher costs of using larger animals. Biomedical research is very expensive, and animal experiments add significant costs. And the advancement of stem cell research, being able to replicate diseases without needing animals, or being able to create mini organs with them, are so important in reducing animal testing and diverting where the money goes. As expensive as it all is, investments, especially in stem cell research, bring about so much potential in truly advancing science and medicine. We certainly didn't arrive at this time filled with investments and excitement easily. The journey of stem cell research dates back to 1950s. During World War II, it was discovered that high doses of radiation could damage bone marrow and blood-producing cells, leading to anemia and immune system disorders. Using mice, researchers then found that bone marrow transplants could restore these cells and fix the problem. In 1956, the first human bone marrow transplant was performed, but the patient died after 5 days due to an immune response. The whole issue of tissue compatibility wasn't well understood just yet. And the question of how exactly this treatment could work remained. The answer came in 1961 from the University of Toronto by Ernest McCullough and James Till, who discovered multipotent hematopoietic stem cells in the bone marrow that could transform all blood cells. This breakthrough started the book on stem cell research. Both McCullough and Till have statues at the university, as well as a stem cell conference in Canada named after them to recognize their discovery of transplantable stem cells. As research advanced, the role of the immune system was better understood in transplants, and in 1968, the first successful tissue-matched bone marrow transplant was performed. In the 1980s, embryonic stem cells were obtained from mice and used in research, and in 1998, the first human embryonic pluripotent stem cells were isolated from surplus embryos of in vitro fertilization IVF treatments. This did raise some moral concerns about the value of human life. Did life begin at conception, or did it begin past the stage of pluripotent stem cells? Did the embryo have a soul? Could it feel pain? Was destroying it the same as taking a human life? The other side of the argument defended that the embryo had no consciousness, and the stage was way too early for there to be a correlation to human functions. That if it weren't for being used for research, the unused embryos would be discarded anyway. But of course, the topic always remained controversial for those who support that life begins at conception. Not to mention the ethical issues surrounding animal research, considering animals were and still are raised in labs simply for the purpose of obtaining their stem cells by euthanizing the mother and her unborn pups. I also worked with animals, and I want to acknowledge that animal euthanasia is a very painful reality for all scientists, but they are crucial to medical research, and every treatment that we trust, from painkillers to advanced therapies, has been tested on animals. As tight as the regulations and ethics behind animal testing have become, I think I can speak for all scientists that we are all looking forward to the day when our molecular and cellular techniques will advance so far that we will no longer need animals to strengthen our evidence. The advancements in organoid research, for example, where we literally grow organs in the lab, have been incredible to understand how we can reduce the use of animals. All I ask is that next time you take even an Advil, do recognize that it was first tested on hundreds, if not thousands, of animals first. Science progress happens step by step, and often this can feel very slow. But the research we're doing today is just the beginning of what could be future treatments. Stem cells are not a magic wand, but in the right hands, with the right questions and the tools, they shape a key that is already redefining boundaries of medicine and science. It was thanks to IVF that we first got our hands on human embryonic stem cells, and now stem cell technology is starting to revolutionize IVF itself. Very recently, in December 2024, fascinating news came from a biotech company in the US called Gamito. Their protocol reduces the IVF hormonal injection period to merely 3 days, rather than up to 14 days of daily injections, to then mature the collected eggs outside of the body using cells derived from stem cells to mature and grow them, followed by fertilization, implantation, and carrying to term. And now they officially have a baby born from this process, which is truly incredible. Once upon a time, IVF was deemed crazy. Now the boundaries of what is possible change every year in science. Who knows what the future holds? That's officially the end of the first episode. Thank you for tuning in, and if you enjoyed this, please do like and subscribe. Santé!